All right, that's my cue for starting the session. Um, Welcome to ARO seminar series that's centered around the topic of vestibular system. Today, we are delighted to have Dr. Kathleen Cullen as our speaker. I am Soroush Sadaghi, and I will be your moderator for today's webinar. Before starting um, today, during the presentations, please, if you have any questions, use the chat function directed at me, if you remember, as the moderator to avoid interrupting the presenter. After the presentation, in addition to using the chat function, you can raise your hands or to unmute and directly ask your questions. Any questions that are not addressed during the session will be addressed by the presenter in the follow-up email. The closed captioning for this meeting is available for use, and this session is being recorded and will be made available for additional viewing. If you have any technical difficulties, please email headquarters@aro.org for assistance. The email will be in chat, so you can use it. As a little background information for Dr. Kathy Cullen, Kathy Cullen is the Raj and Neera Singh Professor in Biomedical Engineering and Director of the Center for Hearing and Balance at Johns Hopkins School of Medicine and Whiting School of Engineering. She also holds joint appointments in the Department of Neuroscience and the Department of Otolaryngology. She is the head of neuroengineering group within the Department of Biomedical Engineering. Dr. Cullen's research has significantly advanced our fundamental understanding of how the brain encodes and integrates self-motion information during our everyday activities. Her lab spans the interdisciplinary fields of neural engineering and neuroscience, and she has always been at the forefront of using the novel computational approaches and neurophysiological methodologies. The overarching focus of her research program is to understand how the brain creates neural representations of our self-motion to ensure the maintenance of balance and posture, as well as accurate perceptual stability during our everyday activities. In addition to her research activities, Dr. Cullen has made extensive contributions to the broader academic community, both nationally and internationally. She currently serves as the president of the Society for the Neural Control of Movement. During her tenure as vice president and now president of NCM, she has successfully achieved gender parity in conference presentations, increasing the representation of women from 10% to 50% to align with the society's current composition. Dr. Cullen has been also an active member of the Scientific Advisory Board for the National Space Biomedical Research Institute, which works with NASA to identify health risks in extended spaceflight. Dr. Cullen currently serves as the steering committees of the Whiting School of Engineering Senate, Kavli Neuroscience Discovery Institute, and National Academy's Decadal Survey on Biological and Physical Sciences Research in Space for the next 10 years. Dr. Cullen earned her PhD in neuroscience from the University of Chicago in 1991. To date, she has published over 160 papers in the fields of neuroscience and neuroengineering, and is frequently invited to write reviews on the vestibular system in respected journals such as Nature Reviews Neuroscience. She has also served as a domain expert for well-known media sources, including the Washington Post, Scientific American, CNN, PBS, Wall Street Journal, CTV News, and NPR. In addition, Dr. Cullen has received awards for research contributions, including the Hallpike-Nyland Medal of the International Barney Society for outstanding contributions to basic vestibular science, and Sarrazin Award lectureship from the Canadian Physiological Society. On a more personal note, I have known Kathy for about more than 20 years now, and she has been both a mentor and a friend over this time. I spent about 7 years in her lab at McGill University in Canada for my PhD research. And I have many great memories from that period. And some of the people in the lab are still very good friends from that time. In addition to everyday experiments, Kathy was also involved in all aspects of education of students. So she's really a pedagogue in that sense, in my mind. And for example, one time after I had practiced my talk a few times in one day with her, I worked on it overnight, and then she wanted to listen to it again at 7 in the morning before my 8 o'clock presentation. So you can tell that everyone had to work hard in the lab, but she was there right beside us, with us. And without further ado, it is my pleasure to welcome Dr. Cullen, who will be presenting her work titled Vestibular Analysis of Head Motion Implications for Perception and Action. Please get it.
Thank you so much, Soroush, for this kind introduction. And thank you to ARO for the invitation to participate in this series. I've really enjoyed watching the earlier presentations by Larry and Jay, and I'm looking forward to talking to you about the ongoing work in my group today. So I'll share my screen and okay, hopefully everything looks good to you. Is that good, Soroush? Yep. Okay, so work in my group is really focused on vestibular processing, as Soroush mentioned. And I'm just going to point out a few obvious things, but I guess points to bear in mind as I go through my talk. So the first point is that in everyday life, our sensory systems are simultaneously activated by stimuli that result from changes in the external world, and these are referred to as X-afferents, as well as stimuli that result from our own motor actions, which are referred to as reafferents. And our ability to distinguish external sensory inputs from self-generated sensory inputs is really essential, both for our perceptual stability, like did I do that or did something happen to me, as well as for accurate motor control. And work in our lab, both recent and past, has really focused on understanding how vestibular information coding or how vestibular information is coded during active behaviors, and specifically the mechanisms underlying the computations that distinguish vestibular X-afferents or externally applied vestibular inputs from reafferents, self-generated inputs. So as most of you know, or all of you know, the vestibular system comprises 5 end organs, the 3 canals that sense angular head rotation, the 2 otoliths that sense linear head motion. Between these 5 sensors, we encode 6 dimensions of motion, 3 rotational and 3 translational axes. Most of what I'm going to talk about is rotating the animal so that we're activating the horizontal semicircular canals, but based on what we know, it's consistent across all degrees of rotation as well as translation. So it generalizes. It's just easiest to rotate animals and activate the horizontal canals in that way. As far as the function of the vestibular system, once you've activated your sensors and you have the information going through the central pathways, as everyone probably knows, the vestibular system generates the vestibular ocular reflex. And for some people, that's really been a primary focus. I think you've probably seen videos like this. So this just shows somebody walking down the street. This is actually back at McGill, walking down St. Catherine Street. One of my postdocs with the GoPro on his head that doesn't have compensation for motion. And this is just normal walking, and you can see how unpleasant it is as far as sort of the retinal blur you get. With every footfall, there's a lot of pitch, head movement, and you get this kind of bouncing around. And obviously that's not how you see the world, because you have a vestibular ocular reflex that generates equal and opposite eye movement to head movement to help keep gaze stable, and it works much faster than the visual system can. In addition to that, the vestibular system generates postural control through vestibular spinal reflexes, and also through thalamic cortical pathways is important for our perception of self-motion. So by integrating information from vestibular system with visual inputs and proprioceptive inputs, you can put together a perception of how you're moving through space. And I'll talk a little bit about that towards the end of my talk. So I've broken my talk into 3 main sections. The first question is, how does the vestibular system encode natural self-motion? And this is going to be sort of almost an engineering style approach to vestibular processing, kind of old-fashioned coding, using some of the computational approaches that Soroush mentioned have been very important in the way I've approached this system. So I initially trained as a sort of electrical engineer, and got more into neuroengineering as my undergraduate evolved. And, the prevailing approach in vestibular and ocular motor systems initially had been really based on linear systems analysis. And this approach has given us a lot of insight into how central pathways basically transmit and process information in order to go from sensory input to an appropriate motor output. So in this approach, you have a sine wave input, which could be a head rotation, for example, and what I'm going to discuss. And then you have some sort of transfer function by which, depending on the frequency, you change the gain and the phase of the output. And the output could be, say, an eye movement if you're looking at the VOR. It could be an afferent response if you want to understand how afferents transduce head velocity information, head motion information. Could be central neurons. So you can go with different nodes along your pathway and look at the response to different frequencies of sinusoidal stimulation and look at the change in gain and phase as a function of frequency. So this has been a very popular approach. So we've done this with afferents. This is work actually Soroush was involved with during his PhD. So we went in and our primary animal model, I should mention, is rhesus monkeys. We have validated, because people like mice now, most of what we've done in mice as well. But what I'll primarily describe is monkey work. So we went into the nerve actually through recording chambers mounted on top of the head. So these are electrodes that are going deep into the brain and hitting their nerve right around the time it comes out of the temporal bone into the brain stem. For the initial recordings, we used glass micropipettes. We've also used some tungsten electrodes to do this and record from individual afferents. And what we can do is sinusoidally rotate the animal if we're recording from, say, horizontal canal afferents on a vestibular turntable. So we're rotating the whole animal en bloc, and he's just rotating back and forth in space. And then we can look at the afferent firing rate. And we can go in and record from both regular afferents and irregular afferents and look at how they respond. I'll just mention, people have been here for other talks in this series, that if you go into the nerve, there are two classes of afferents, and they're called regular versus irregular based on their resting discharge. Regular afferents are regular. They have a very specific inner spike interval between each action potential. On the average, about 10 milliseconds. They fire at about 100 hertz on average in monkeys. And then you have irregulars that have a much more broad distribution if you look at their inner spike intervals, because they're more irregular. And this is in the absence of any motion, just the animal sitting stationary. And this is sort of an approximation, but in general, you have two types of hair cells. You have type 1 hair cells that have been discussed, which have the afferent that comes in, actually hugs around the whole cell body in this sort of calyx-shaped structure. And then you have the type 2 hair cells that have a more conventional sort of bouton synaptic transmission profile at the base of the cell. And on average, the type 1 hair cells are more strongly innervated by irregular afferents and the type 2 by regular afferents. There are also, of course, geographical differences across the neural epithelia, but I won't belabor those. The main point really is that this type 1 hair cell evolved more recently. So, animals that live in water don't have type 1 hair cells. It's only once animals sort of moved onto ground that they evolved and left an aqueous environment. So it's very interesting to think about why this second channel evolved. So it's something to bear in mind as I go through my talk. So what we can do is rotate the animals back and forth at different frequencies. We have here 0.5 hertz and here 15 hertz. Again, these are horizontal rotations, so we're activating afferents that innervate the horizontal semicircular canal. And you can look at the example regular on the top and irregular on the bottom. And the main thing to see is that as you increase the frequency of stimulation, even though the amplitude is comparable in terms of head velocity, you get an increase in gain. And this gain increase is even more striking for the irregular afferent than for the regular afferent. And you can fit these responses. They're really beautiful. We can see they're encoding the head stimulation very well by fitting a resting discharge and then a gain term. And this little TD here is really just a way of representing the phase. So it's a phase shift in time. So when you do this sort of analysis, what you see across this whole frequency band, so going up to about 16 hertz in this example is that the regular afferents here in blue, sort of the older channel, presumably, have lower gains across this whole range of frequencies than do the irregular afferents. And I should note this range that goes up to, say, 16 or maybe 20 hertz is the physiologically relevant range, and I'll talk a little bit more about that later. But if you put a MEM sensor on a person or a monkey or a mouse and see how they're moving through the world, most of the power is below 20 hertz. So really, that's the range that the vestibular system is trying to encode. Okay. So this then, sort of begs the question, well, if these irregular afferents are so, peppy and mighty, what's the point of hanging on to these regular afferents? And to answer this, I'm just going to put up a video that Soroush actually made years ago, which I think is really good, and I use whenever I teach this to people. So what this shows, the yellow trace that you're going to see running out as a function of time is head velocity. But again, this would be a horizontal canal afferent, and the magenta trace is a regular afferents firing rate. So you can see here's the baseline discharge. It's nice and regular, around 90 hertz. There's the head movement. There's the afferent response, and it's really beautiful. That one little blip you see there is probably us mistriggering more than the neuron not following the stimulus perfectly. So if you had one regular afferent, you could do a really good job of guessing what the stimulus was here, right? So if we go to the next example, and this is an irregular afferent, you can see in the absence of any stimulation, there's much more noise, right? And that's the irregularity. And now during the stimulation, there is modulation, but again, it's a much noisier sort of modulation than we saw with the regular afferent. And then again, when you are stationary, or the monkey's stationary, we get this sort of noisy profile again. So I think that really helps to think about this question of, why do we have these two channels? This channel has higher gain, but just looking at it, it's noisier. So that led us to think about how these two channels are actually encoding natural self-motion stimuli that are generated in natural conditions. So this work was done primarily by a postdoc, Jerome Carriot, and a graduate student, Mohsen Jamali, in my lab. This was kind of exciting. It was right around the time that these MEMS or IMUs were becoming available, were getting smaller. I happened to talk to some people at ST Microelectronics. They had a small eval board that we could actually hook up to a cell phone battery and a data card and mount in a kind of at that time, tiny, at this point, kind of big module that we could attach to people's heads. And we had them running around the city of Montreal. We had them on buses and trains and doing all kinds of things, just trying to get a sense how people really move. And this was in part initiated because Mohsen was going to do some analyses of otoliths, and we're looking through the literature trying to understand what kind of linear motion profiles do people generate in everyday life, and we couldn't really find anything. There was some work on treadmill, but really nothing about how people were moving. So it was really, fortunate in terms of the timing of the technology. So what did we find? We found through looking through all these different types of behaviors that we recorded that, a lot of time you're not moving very much, like all of us are just sitting and right now and listening, or I'm talking. But you can reach very high intensity movements up to 8 Gs, very transiently, 15K degrees per second in terms of rotations. The distributions of our movements are highly non-Gaussian. They're very peaky in the middle. You can see that down here. The middle would be 0 because you're not moving, but you can really reach these high intensity movements. So these long tails. Significant power up to 20 hertz is already mentioned. And for people who know anything about natural statistics of natural stimuli, if you look in the auditory systems or visual systems and look at the statistics of natural sounds or natural images, and you do a log-log plot of power versus frequency, you get a nice line, a nice linear relationship. And we don't see that in terms of the vestibular input. There's always a break somewhere between, say, about 2 and 4 hertz, where you have a lower slope for low frequencies and then this higher slope at other frequencies. In separate work I won't talk about, we've looked at how central neurons deal with that input, and we found that central neurons actually displayed whitened responses that are appropriate, or responses that are appropriate for whitening responses to these stimuli. And that gets in this whole world of optimal coding. Okay. So we've also done this. Of course, we don't work in people, we work in monkeys and a bit in mice. So we actually looked at this a bit in monkeys and mice and found that fortunately for us, if you look at the natural statistics of how monkeys move, and we had them in big playroom with cages and other monkeys, and we were able to miniaturize our little module even more and just see how they moved, swinging, eating bananas, doing things monkeys do. And their statistics looked a lot like humans, and also had that kind of break in about the same place when you do that analysis to see if the movements follow a power law. And we also looked at mice, and there were some slight differences in mice, but generally quite similar. So one thing that we pull out of this is that if you look at what people have done in my field, and this is sort of, if you have a particular tool, it can be great because you can make strides in the field, and linear systems had been really wonderful in understanding, dynamics and building models and looking how the biomechanics are then, further processed, the biomechanics of the canals shape the way afferents encode information, but then you have channels and other processes that further shape the dynamics. So linear systems analyses have been very useful in this way of trying to understand how different parts of the processing and transmission, ultimately shape the dynamics. But in order to use linear systems, you need to stay in a linear range. So we've almost been taught in the vestibular world to stay in this linear range, so we could use this tool. But the issue is if you actually look at the range of movements we generate, we spend a lot of time well outside of that linear range. So that suggested perhaps we should look at some other ways of looking at information coding in these pathways, and so that's what we did. So we went back and recorded from regular and irregular afferents, and we could use natural head movement stimuli. So we could take replays of the movements that the monkeys were actually generating as they were running around in these play cages or playrooms, and play them into our motion device and see how the afferents responded to them. I'll just take a moment to mention that you can make a prediction, and this is kind of obvious from the movies I showed you, those little videos. So if you look at a regular afferent when the animal's at rest versus an irregular afferent when the animal's at rest, and you can see the interspike interval here is, really clean. It's, neuron fires every 10 milliseconds if you look at the ISI distribution. And here you have a much broader distribution for the irregular afferent, right, as you would expect. And then you can take the power spectrum of those resting discharges. What you get for the regular afferent is a nice peak at 100 hertz, which is consistent with its firing at 100 hertz. And then you get the subsequent harmonics at 200 and 300 hertz, right? So just what you would expect. If you do the same thing for the irregular, you get this much broader distribution, which is consistent with the fact there's less patterning in the resting discharge. And then this gray bar represents the range over which these neurons or these afferents are trying to encode head motion, right? That's the physiologically relevant range that we know that head movement is generated in. So we know that most of the power is below 20 hertz. So what this suggests is that, within this range, the lower power in the regular afferent's firing rate predicts less noise. There's less noise to overcome for coding something that's being experienced, compared to what we see for the irregular, where there's a lot of just baseline noise there over which you're going to have to then do your encoding. So that was our prediction. So what did we do? We went in and we took these more complex stimuli. So the gray shows the head motion that we're applying for a regular afferent. The black, actually I'll go down to the bottom. The sort of raster at the bottom is the action potentials fired by the regular afferent. The black trace represents what we call a spike density, where we took a Gaussian function and just convolved it with each spike. It's a way of representing firing rate. And if you compare the gray and the black traces, you can see that the firing rate actually, does a pretty good job for this regular afferent of predicting what the actual stimulus was. And then you can do even better. Rather than just using a Gaussian, you can estimate the optimal linear filter that you can do the convolution with this spike train. And that's what this dashed blue line is. So this is the convolution with the optimal linear filter, and we call that the stimulus reconstruction. So you can see you can do a really good job with just a single afferent. Again, this is for head rotations of reproducing the head motion stimuli. If you go through that same process for an irregular afferent, what you see here in the red dashed line is the stimulus reconstruction, the best one you can do for that irregular afferent, going through that same process. So the firing rate is here, the gray is the head motion, and you can see it's a very poor reconstruction, right? So what it suggests with a single irregular afferent, you can't predict as well the actual patterning of the stimulus. So what this shows, and this is consistent across the population of afferents we recorded from, regular afferents have larger coding fractions than irregular afferents, so it follows that regular afferents carry more information in their spike trains. And this is work actually that Soroush did. Here you can see this really nice response where the regulars have much more information across the entire frequency range than the irregulars until really this very end. There's only a slight crossover at about 20 hertz. And again, just by comparison, if you're looking at terms of gain, sort of the traditional way that people have always looked at vestibular coding, what you see is that, again, the irregulars always have higher gain across that same frequency range. So the other thing we can do, so what I focused on here is rate codes, but as people who study the auditory system know, there are also timing codes. And given sort of the common evolutionary origins of the vestibular and auditory systems, I'd always been curious to know if, in fact, there was some sort of timing code that was being used in the vestibular system. So you can just see these are two windows, right? So the number rate code is the number of action potentials during a given time window, and how does that carry information? So, in both of these examples, it's, here and here, if we just look over this whole window, it doesn't matter if you're looking at the top and bottom, there'd be 4 spikes for that interval, and the same for here. But obviously on the right side, in that example, there's much more precision within that given time window, and that would indicate potentially a timing code. So how did we look at that? So here to do this, what we did is a replay. So we took epochs of our natural stimuli and played them over and over again. So here's an epoch of the natural stimulus recorded from a monkey in his playroom, then played into our motion table. And what you can see for an example irregular afferent, we play this over and over again and record multiple times from the same irregular afferent, and then do the same thing for the regular afferent. And just by looking at it, from a distance, you can see there's some sort of banding that occurs in the case of the irregular afferent, and some structure that you don't see in the case of the regular afferent, even though we know that ultimately the regular afferent is encoding with higher information, right, than is the irregular afferent. So irregular afferents displayed what looked like lower trial-to-trial variability in the regular afferents. So how do we quantify this? So what we can do is take, instead of just one stimulus and playing it over and over again, multiple stimuli. So this is the blue is stimulus 1, red is stimulus 2, green is stimulus 3, and then record from the same afferent for all these. And we don't just stop at 3, you can go to, say, n equals 20 and do this multiple times. And look at the banding. And then what we can do is use, there are a couple of metrics you can use in order to determine is whether or not there's some sort of spike timing precision. These are metrics where you take, for example, the responses to each stimulus. Let me go back. I think it's easier if I just do it this way. So what we're going to do is train a classifier and take for each stimulus the top response, and hold those aside, and they take all the other responses, and you can think of kind of tossing them in a paper bag and then pulling them out one by one. And you can use a metric that has sort of a cost. You can move spikes around, you can drop spikes out. And so you have each of these top 3 responses as your template. You pull a random response out and say, which is it most like? And you basically can realign things until you've got it kind of aligned as well as possible. And the higher the cost of doing this alignment, the poorer it is as far as a fit. And you can do this using a couple different metrics there, but for our analysis, they came out with similar results as the Van Rossum metric or the Victor Purpura metric. And ultimately what you do is you assign the response as being listed by the stimulus that gives rise to the template that's closest to it. So it has the lowest cost. So when we do that for the irregular afferents, we end up with a performance that's close to perfect. And you would bootstrap this. You wouldn't just always pick the first response. You would do this over and over again, taking all responses and bootstrapping it properly. You also can do this at multiple timescales in each of these metrics. And then if we do the same thing for the irregular afferents, we find much poorer performance. So that means if you went through that exercise, you'd have a harder time based on the response, pulling a response out of pulling out another response and matching it to the correct stimulus. And if you do this over our entire population of afferents, what we found, again, was really excellent performance, ability to identify the correct template for our irregular afferents. And that occurs at a time scale much higher frequency than anything that actually is in the stimulus itself. And then in contrast, if you look at the regular afferents, they're much poorer, which means they don't have as much spike timing precision. And they actually peak at a frequency or a time scale that's really close to the end of the frequency range of the stimuli we were applying, which would be consistent with about 20 hertz. So what this has shown us is there are two distinct information channels in early vestibular processing. One that uses a rate code, which would be the regular afferents, as opposed to the irregular afferents, which are really quite poor at transmitting information through a rate code. But then if we flip it and look at temporal coding, the temporal coding in the irregular afferents is much better. We've since gone on and started to record from central pathways to understand how these rate versus temporal codes are used within central pathways that generate things like the vestibulocular reflex or vestibular spinal pathways. These VO neurons are neurons that we know are involved in sending projection sound down to via the vestibular spinal pathways to the spinal cord to generate postural responses, and also project up to the vestibular thalamus, in particular the VPL, ventroposterior lateral thalamus, in order to send information up to cortex and areas like PIVC that are involved in perception. So what you can see is these VO neurons, somewhat surprising to me initially, but the more we look at it, it becomes quite interesting, look more like irregular afferents. And that's a bit consistent with work that people like Jay Goldberg and Steve Heistein did, showing that irregulars have a tendency to more strongly project to these VO neurons. And they also show better temporal coding. So what we think is that this pathway is set up to generate kind of a burst or a more kind of synchronous spike timed input to the spinal cord to overcome the inertia that you need to generate postural control. We've done similar experiments on regular neurons versus PVP neurons, which we know are involved in vestibular ocular pathways, and they seem to be a little bit more similar, which is consistent with the idea that for the vestibulocular reflex, you might need better patterning in order to really have better preservation of the patterning of the head movement in order to generate appropriate gaze stability. Okay. And I added this slide in going over things last night because I thought it might be helpful for people who come from a more auditory background. So the spike timing we see is consistent with the fact that when you actually, rather than using a naturalist stimuli, use sinusoidal stimulation, that we actually do see phase locking, and that the phase locking is greater for irregular afferents than it is for regular afferents. So you can just see an example there. And the greater the amplitude, the more striking that phase locking is. Okay, so just to tell you kind of what I've told you, or review what I've told you so far, we have two distinct information channels in semicircular canal afferent coding, which is what I focused on in all the slides I've shown you so far. Regular afferents are low gain, whereas irregular afferents are high gain. And we knew that before, but we extended the frequency range over which we tested this to the full natural range. Regular afferents are high information, and they use a rate code. And as I mentioned, they look like they're this information is transmitted relatively faithfully onto the VOR pathways, the central pathways that generate the VOR, whereas the irregulars are high gain yet low information, and they use a spike timing code, and that seems to be then faithfully transmitted onto the vestibular spinal and pathways. And we're actually looking more at the thalamic cortical pathways. They seem to be a little more complex in how they're integrating this information. I won't belabor this, but we've also looked at otolith afferents, and it's a little different. Regulars are low gain. Regulars are high gain. That's the same. These guys are actually, the irregular otoliths are so sensitive that their gain overcomes their noise. So they actually have higher information than do the regular otoliths. But the regular otolith afferents, right as you get to really low frequencies and are looking at static changes in orientation, actually have better discrimination. And then again, these guys have a rate code, being the regulars, whereas the irregulars have a spike timing code. Okay, so I'm going to move to the second part of my talk, which is how everything I've talked about so far is natural motion, and how do you encode across, with higher amplitude movements and nonlinear codes like spike timing codes and looking at information. And we looked at natural behavior, but ultimately the animals were not generating the behavior. We quantified the natural behavior, and then we took the behavior and played it into our turntable. But what if the animal's actually generating, or you're actually generating, an active behavior? How are the vestibular pathways incorporating that information? So the first question is, why is this an interesting question? And it's been well known back from the days of Helmholtz that, for example, if you tap on the side of your eyelid, you get a little shift of the world. But if you make saccades that'll move your eye even quicker, you never see this sort of disjointed, jumping of the world. And that's because in the second case, when you generate a saccadic eye movement, your brain has access to that premotor or the motor command to generate that eye movement, and it can use that to kind of stitch things back together. It's also important in the context of accurate motor control. So in the vestibular system, the same central neurons that receive direct afferent input also project to motor centers that control both vestibular ocular and spinal reflexes. And if you consider vestibular spinal reflexes, they're essential for providing robust postural responses to unexpected vestibular stimuli, but they can be really counterproductive when you want to move through the world purposefully. So this is what I show here. It's somebody walking down, an icy sidewalk on a hill, and they're about to slip, and they're going to get a big vestibular input that they don't expect, and hopefully they're going to use that to keep themselves upright, right? And that would be unexpected vestibular input, or X afferents. And there's somebody moving purposefully and maybe experiencing very much the same vestibular inflow, but she's not going to want to be mounting vestibular spinal reflexes to keep her upright, because that would actually, or keep her stationary, that would make her fall out of her jump, right? And she has, done this jump many, many times, and this is something, that she expects. She has a, very strong sense of what kind of inflow she expects, and she's going to use that to know if she's doing her jump correctly or not. So these are two very different conditions from the point of motor control. So that's why, why would you want to make the difference? The next question is, where could you make the difference, or where could the brain make the difference? And the nice thing about vestibular pathways is they're very streamlined. So we have afferents coming from the end organs, right? Canals and otoliths, projecting to central neurons in the vestibular nuclei. And I already referred to these. The VO neurons project to spinal cord and to VPL, ventral posterior lateral thalamus. And then we also have the VOR neurons, and they project directly to the motor neurons that move your eyes. So you have basically central neuron that projects either right down to the spinal cord or right to the motor neurons that move your eyes. So 3 neuron arcs in their most simple form between sensing and behavior. So the first question is, well, what happens at the afferents? And that may seem like a silly question, because the afferents are bringing information in. So the question is, why would they be different? But we know there's a vestibular efferent system. And at the time at which we did the initial experiments, the reigning hypothesis, or a reigning hypothesis of the vestibular efferent system, is that it was involved in preventing the vestibular system from cutting out. And what it would do, during very transient purposeful head movements, would pull the resting discharge up and reduce the gain, so you were less likely to saturate your sensor. So that's what we actually expected to see. What we did is we had the animals sitting on in their monkey chair. They would generate active head movements, so we freed their heads. And then we could do a replay of that active head movement right into the turntable. So they moved their heads back and forth like they're shaking their head no, and then we would replay that into our turntable so that the head would move the turntable so their head would move in space the same way. And what you can see right away is this afferent responds the same way, regardless if the movement is passively applied or actively generated. We can fit a little model with a bias or resting rate, and then the head velocity and acceleration terms are basically ways of representing the sensitivity and the phase. And when we do that, what we see is that regardless if the animal is passively moved or actively moved, there's no difference. The response, the sensitivities are comparable, and there's no difference in background discharge. So there's no increase in background discharge is predicted or decreased in the sensitivity. In talking to some of my colleagues, people said, well, you weren't looking at, just head movements. You didn't look at locomotion. You need to have the animals moving faster. So most recently, we actually had our monkeys running and walking along a linear track. We could keep track of their movements using something called deep lab cut. So we knew where they were in their gait cycle. We could record their head movements using IMUs and look at how the afferents are actually encoding information during these more natural running and walking protocols. And the organization of the data is pretty comparable. We can do a passive pitch that basically, for say a vertical canal unit afferent, have the animal moving its head up and down where we do it as experimenter, and then he does it himself as he's running or walking. And what we found again is there's no difference in the bias or the sensitivity either during walking or running as compared to passive. So the good news is that this means that everything I told you in the first part of the talk about how vestibular afferents encode information is true regardless if the monkey moves himself or we moved him. So everything I told you about gains and information coding and rate versus spike timing codes holds. The other good thing, and I'll go through this pretty quickly, is this is a little bit of a detour, but is it has important implications for vestibular prosthetics. So one of the reasons I moved from McGill to Hopkins was I've had longstanding collaborations here with people like Charlie Della Santina and Lloyd Miner when he was still here. Charlie has ongoing clinical trials with patients with profound bilateral vestibular loss. About a dozen patients have been implanted. So these are people who have bilateral loss. They're implanted on one side. And if in fact you needed a different mapping between head motion and stimulation of the prosthetic, would be complicated, right? If you needed a different mapping for active movements versus passive movements. So what this suggests is we can use the same mapping regardless if the person is generating their own movement or they're being moved through space, which is helpful. The other thing we can do is take all the information that I told you about and try to use it to inform a more biomimetically inspired prosthetic. So the way the prosthetic works is this is currently replacing the three canals. So it doesn't replace otolith function, but is focused on the three canals. They're stimulating electrodes in each of the canals in the ampulla. And what you can do is record three-dimensional head rotation using a gyro, map that head rotation into the three canal planes. And then once it's mapped into the canal planes, you know how much motion is in each of those canal planes, but you still need to map it into a pulse train. And what we can do is take advantage of what we know in terms of the way that afferents fire. So we can take those transfer functions, the gain and phase as a function of frequency, and use that as part of the mapping. But the other thing we can do is also note that as the head movements you generate in everyday life tend to saturate. So in order to do this, we need to also include the static nonlinearity that describes that saturation and the cutoff that happens when you're in the off direction. So early vestibular processing, as I already mentioned, is not linear over the natural range of head movements we generate, and people have developed in other sensory systems, and we've adapted this to the vestibular system, these linear nonlinear models that include a static nonlinearity. And once you include, the idea is that if you don't have that, you predict negative firing rates, which the neurons can't do, and that's kind of one of the reasons that people had been keeping in this small range when they were probing the system. But if you account for this nonlinearity, you can do a really good job of representing how the afferents fire. So this is the current kind of mapping structure approach we're using, and we actually have monkeys implanted with a prosthetic to try to understand how to best, how different mappings both affect the precision of sensory motor processing. For example, we can look at the vestibular reflex, and we found that if we use a mapping that approximates afferents that have regular responses, so regular afferents, we do the best job of reproducing the VOR timing that you would expect in a normal animal. And we're now also applying this in the case of looking at animals in a posture platform and during walking. The other thing we can do is go in and look at how the stimulation protocols that people are currently using in the clinic affect neurons. This is work I don't have too much time to talk about today, but we've found when we go in and record either from the neurons in the vestibular nuclei that comprise the VR pathways or vestibular spinal pathways, that we can get these effects that were sort of unexpected. So typically, because these are unilaterally implanted, there's a baseline discharge that's set maybe around 200 pulses per second, so you can go in two directions. And we found that, in fact, this nerve activation actually induces long-term depression at the vestibular afferent central neuron synapse. That adaptation actually hangs around for a long time, and it's not a good thing because it actually reduces the efficacy of transmission by about 50%. So in this way, we're using the ability to record from these pathways as a good indicator of what are the current bottlenecks in fully restoring function in people, and what maybe can we do for the next generation of prosthetic. So that was a little bit of a tangent, but I think it's an interesting point, because now that we know regular or afferents encode information the same way regardless of whether movement is active or passive, we can kind of move forward and use these more biomimetic approaches. Okay, so again, we don't have a lot of real estate here. We just have 3 neurons, say, for example, vestibular spinal pathways. It's afferent central neuron, then projections down to spinal cord. So if we go to the central neurons in the vestibular nuclei, what do we see? And we see something that's quite different. Here you have active head movements, and here you have the replay of the active head movements. And you can see when the movement's active, this central neuron doesn't respond very well, but it does to the same movement when it's passively applied. And we can do another experiment where we actually had the animal rotating on his platform while we let his head free. So that's what you can see here. So the dashed line represents us applying a sinusoidal rotation back and forth. The black line up here represents his total head and space motion that we can measure. If you subtract the dashed line, which is externally applied, from total, you get this black line down here, which represents his active head and space movement. And what you can see really clearly is, we're applying a sinusoid. He's generating something more complex. This central neuron is only encoding the externally applied part of the head motion. So it's a pretty profound suppression that we're seeing. And this is at the first synapse, right? The first central synapse. And just to make it really clear, if we're recording from an afferent, right, it would encode total head and space velocity, right? Because afferents encode information in a context-independent manner. We did another series of experiments. This was done by my postdoc at the time, Jerome Cario, and Isabel Macros. The question is, what happens if you actively versus passively redirect your head relative to gravity? And we found that the gravity-related responses of central neurons mediated via the otoliths are also canceled during head movements. Okay. And we're currently now looking at how vestibular processing is modulated during even more natural behaviors, such as locomotion, as I already showed you for the afferents. Okay, so I'm just going to finish up by talking about the cancellation signal itself and how it's generated. And that's, we've demonstrated that afferents encode information during active and passive movements in a context-independent way. I've showed you the different types of coding that's done in the regular versus irregular afferent pathways. And then I additionally shown you that you have this profound suppression at the first synapse in the vestibular spinal pathway. And I should note, we don't see this in the VOR pathway. The VOR pathways act in a different way, and I'm happy to take a question about that at the end. So question is, how is this cancellation signal generated? Because the afferents encode information, but the central neurons have this context dependency. So you need to generate a cancellation signal for when movement is actively generated. And I just note that we see this for linear motion as well as rotational motion, repositioning of the head relative to gravity. And we also see this in mice. Okay. So the hypothesis we have, and we've done lots of experiments and I'm happy to discuss them, but I'm going to give you a short version of it today, is that we see cancellation of vestibular reafferents when sensory feedback matches that expected as a result of the motor command that was generated. So it's kind of like the skater's done something many times, and she has an internal model in her brain of the sensory feedback she expects for a given motor command. And this starts to sound like motor learning, because the idea is in motor learning, the brain, in particular the cerebellum, computes an estimate or internal model of sensory consequences of action, which it then compares with the actual feedback. And when you compare this prediction versus the actual sensory feedback, whenever there's a difference, the difference is called a sensory prediction error. So in motor learning, that error is what drives learning. But in our problem of understanding encoding of unexpected motion, it basically allows the distinction between vestibular X afferents and reafferents. So in order to test this, we recorded from cerebellar output neurons, and vestibular nuclei neurons are also effectively cerebellar output neurons because they get direct Purkinje cell input, as does the area or the deep cerebral nuclei is called the rostral fastigial nucleus. And we recorded from both vestibular nuclei neurons, VO neurons, and these fastigial neurons, and they're getting input from an area called the anterior vermis, which we know is involved in posture. This was done with Jess Brooks, who was a graduate student with me, and Jerome Carriot. So the idea, again, here was our hypothesis. You need a match between what you expect and what actually occurred in order to get suppression. And we said, well, if this is true, we should not get suppression when the relationship between the motor command that is generated and the sensory feedback is altered. So to do that, we set up a little learning experiment where we had the monkey sitting in this monkey chair. He's making active head movements, but we could put a resistive torque on that on his head movement. So he's making normal movements, and then we can put this torque on when we want to. And then he has to deal with this torque and overcome it in his motor control in order to get, to generate the appropriate movement and get his reward. So we can run normal passive movement, experiment, normal active, and then we can put the torque on. There's a learning phase where he learns he needs to generate a bigger motor command to make his head movement. Once he's in steady state, we can take the load off every so often in catch trials, and then we can eventually just extinguish the behavior by taking the load off completely. So this just shows the time course of the behavioral adaptation. So you see normal passive and normal active movements, like I've shown you before. You put the resistive torque on and you have early, middle versus late learning. It takes about 50 head movements to get back where he started. And initially he's only moving his head half as much. And then once he's in this new learned state, when we take the load off every so often, these are called catch trials, and you get this big overshooting because he's now updated his internal model for this load. So what did the neurons do? Well, the first two columns you already know. They encode passive motion, and not active movement. So they look, these fastigial nucleus neurons look a lot like VO neurons, and in fact, they look exactly the same even through the learning. And then what we can do is look at through the learning, and the dashed red line represents how the neuron should fire if it coded motion with the same sensitivity encodes passive motion. So what you can see here is that this first column in learning, in early learning, the head only moves half as much, but the neuron fires more than it does in this condition, right? And in fact, it fires as much as you'd predict based on the neuron sensitivity to passive motion. And then halfway through learning, you get about 50% reduction. And then by late learning, it looks a lot like it did initially during normal active movements. And another way of presenting that is here, normalized sensitivity. So the neuron's sensitive to passive but not active movement. But when you make this mismatch by applying a torque, so there's a mismatch between what the monkey does and what he expected he'd get in terms of sensory feedback, it's initially the movement is encoded as if it's passive, and then we get this reemergence of reafferent suppression. And then for all these catch trials where, again, you induce a mismatch from what the animal's expecting, you get encoding as if even these are big active movements, but they're encoded as if they're passive. And you can look in terms of the head velocity error, and they follow that very nicely. So in summary, when the relationship between the motor command to move the head and resulting actual motion is altered, the neural responses are consistent with the rapid updating of internal model that predicts the sensory consequences of self-motion. So this is important for accurate motor control, as I already mentioned. You know, if you're purposely moving through space, you don't want your vestibular spinal reflexes stopping you from doing that, and you want some sophisticated gating. So this is a mechanism by which that can occur. Effectively, when unexpected sensory input becomes expected, the brain internal model is updated to re-enable a distinction between vestibular reafferents and exafferents, as I just showed you. And that's important for calibrating motor circuits. I also had mentioned perceptual stability. I'll do this very quickly. We've looked at the VPL, ventral posterior lateral thalamus, which gets direct input from these VO neurons. Just for those who are interested, we've looked at them a lot during natural stimuli and characterized them in terms of their responses to passive inputs, but I won't talk about that in detail today. But we've also did a comparable experiment where we looked at passive versus active and then combined movements. And what you can see is these neurons, again, selectively encode just the externally applied component of motion and largely ignore the actively generated component. And this is important because, you know, if you're moving through the world, you know, you want to be able to perceive what's unexpected motion. You know, and anyone who's ever experienced an earthquake knows that even that small amount of unexpected movement is a really salient stimulus that you're quite aware of. So you can think of this circuit as keeping track of that sort of unexpected motion at a perceptual level. Just to summarize what I've shown you in terms of passive versus active responses, if you look at the afferents, they're context independent. They respond the same way if motion is passive or active. If you go to vestibular only neurons, they encode selectively passive motion. You get about 30% attenuation. The same with these rostral fastigial nucleus neurons I talked about, which are part of the deep cerebellar nuclei. And thalamus shows actually even more selective coding. You're down to about 20% of the response, so about 80% suppression. I don't think I have time to go through the last part of my talk in any detail, but I just wanted to give you a general sense of what we're doing. So I'll go through this really quickly. So I've showed you how afferents respond in active and passive conditions, talked about different coding strategies. I talked about the fact that we see reafferent suppression at the level of the vestibular only neurons that are the vestibular spinal pathway neurons. And now what we're really trying to do is understand how the cerebellum builds this internal model. So we can build these black box models, and they're very good ways to think about things and formalize hypotheses, but they don't actually tell you how the brain is doing the computations it's doing, right? They're models. So what we've been doing, and this is with Omid Zaberi, who's a PhD student with me at Hopkins, and Robin Mildren, who's a postdoc here, is trying to understand the level of cerebellar cortex. How is the brain actually building these internal models? We can go in with new technology. We now have these non-human primate neural pixel probes that have 4,000 channels across the shank. We can call it multiplex between about 380 at a time and really record from lots of neurons in terms of populations. And we can look at vestibular coding across the Purkinje cell layer, and we find sort of a zoo. There's a lot of heterogeneity in the way that these neurons encode, and they're far less linear. There's really no linear range. Some of them rectify, some of them are V-shaped, some of them are linear, some of them are other in terms of their vestibular coding. And they're also sensitive to neck proprioception, and we can look at that coding. We can build population models now based on this and try to understand how that heterogeneity is coming together to build these internal models. And in particular, we're exploring how these multiple sensory streams being vestibular and proprioception are combined with motor input in order to compute an estimate of expected self-motion. So that's paper that's currently under review, and we hopefully will be able to share soon. The other thing we're looking at is, again, we can build these models and say, okay, you need a match between expected and unexpected, or expected motion and what actually occurred. And the question is, how do you make that comparison, and how do you determine if there's a mismatch, and what are the rules of that comparison? So something else we've been looking at is, how tight does that comparison need to be, and how does that actually translate to the natural variability that you see in movements? So if you generate a movement, there's lack of repetition and repetition. There's always trial to trial variability in the control of movement. So, this would be the animal generating very stereotyped head movements back and forth. There'll be a mean head movement, but we have a lot of variability, both in terms of speed and the duration of the movement. So what we can do is apply systematically assistive or resistive torque within this very small range and kind of probe what is the tightness of this match. So there are two hypotheses. One is you've got this big area which anything within the range of natural variability is encoded as being okay, or a good match, or this other where you've got a more notched sort of comparison going on. And what we found so far is that our analysis looks more consistent with that latter hypothesis too, where we find a gradual reduction in reafferences, even for tiny perturbations. We start to see that we've moved from our match. So I'll stop there. I'll just mention that we focused in the cerebellum currently on the anterior vermis, but as people who are vestibular experts know, there's also the nodulus uvula, and we've been looking in as well in the nodulus uvula during active and passive movements, and have some very interesting and unexpected results that I'm looking forward to sharing with the community. So in summary, what I've told you is that afferents encode vestibular information in a context-independent manner, at least across all the active behaviors that we've looked at, including walking and running, whereas neurons in the first central state of processing preferentially encode passive self-motion, or X-afferents, and that's specific for the vestibular spinal, vestibular thalamic neurons. The VOR neurons are different. Vestibular sensory responses to dynamic self-motion and sensory consequences of gravity are canceled when there's a match with the brain's expectation. And that was the learning experiment I showed you. And the output of the cerebellum reveals this rapid updating of an internal model as unexpected sensory input becomes expected. And the distinction between active and passive vestibular stimulation ensures both perceptual stability and accurate behavior. And we're looking more at these ascending thalamic cortical pathways, and are currently reporting from area PIVC and cortex. And then current work is focused on computations underlying the cancellation of active vestibular input reafferents. Also, how does the cerebellum and or cortex build these internal models? Skip that. This is just, again, to point out the VOR. You don't want the VOR off during active movements, because as I showed you in this initial movie, you want the VOR as you're walking down the street. But I can talk about the gating that does occur in the VOR at the end in the question section. And I just want to thank, of course, all the people. I tried to call out most of the people involved in what I really focused on this talk, but it's a big team of people, including people from my days back at McGill, and then a new team at Hopkins. So I'll stop there and thank people for their attention, and I'm happy to take questions.
Thank you very much, Kathy, for this presentation that summarized a lot of work very nicely. We know we've passed the time, but there's time for maybe a couple of questions if anyone wants to start. Otherwise, I have questions always. So I'll start by the obvious question is, what happens to the active head movement information then? Is it all lost in this pathway? And we obviously need that information for spatial orientation and knowing where we are, and the sensation that when we move our heads, we know that we are moving it. So just.
Yeah, I guess the active head movement, it's obviously not lost because you have a motor command of it, right? So the question is, where do you need it? So you don't need it necessarily in your vestibular spinal pathways, or, during walking you might. So the question is, there's work done in, say, in human using GBS, looking at the way, so people like John Sebastian Blouin use GBS in people, and they look at vestibular spinal pathway responses during gait cycle, right? So during walking, we're not sure. It might be gated in a way that's consistent with, transitions and sometimes wanting that extra postural control and other times not wanting it. But, in terms of, actively rotating your head, you always have that motor command. It's not that it's lost. The question is, where do you need it, right? As I alluded to, when we're looking at nodulus uvula, nodulus uvula is different than anterior vermis. Nodulus uvula gets a lot of direct afferent input. So we made a prediction going into nodulus uvula that we might not see this suppression. We actually did like a poll around the lab. I don't think anyone put money on it, but and it looks like nodulus uvula is different, right? It looks like we're not seeing the same suppression, which is interesting because it seems like almost everywhere we go, we keep seeing this suppression during active movements, right? The question is, how is that then used? And that opens another question, since that information would also be coming through the same, nodulus uvula projects down to vestigial and to the vestibular nuclei where we do see the suppression, but, it's possible that it is in part going to the VOR pathways, which don't show the same suppression, right? If you went through this whole series of experiments I showed you for the VOR pathways, the PVP neurons, they don't, if you're moving purposefully through space and you want to keep your gaze stable, they're doing their job. So it could be that that pathway is helping kind of, realign eye movements and keeping them consistent with gravity for. I don't know if that answers your question. Yeah,
absolutely. Yeah. Larry Hoffman, you had a question.
Thanks very much, Kathy, for that wonderful summary of a plethora of work. So I guess two questions. One is you for obviously a number of years have talked about internal models. Have you thought about how that may be integrated in a Bayesian context? That is, the relationship between your internal model and the Bayesian prior, which again is an instant. Basically, colloquially, it would be represented as an instantaneous update in a state-dependent sort of fashion. And then following up on that, in your work dealing with your animals on a treadmill, is there any evidence, or perhaps even in other work, because I haven't found it, of a potential contribution of descending inputs from descending cortical inputs? In other words, so you initiate a behavior in motor cortex, and whether there's any evidence for contributions of that ability to modulate or to create a state dependence on your internal models.
Yeah, I mean, I guess in terms of, we did try some other things in terms of state dependence and I guess we looked at it from the point of view of trying a different, set of protocols that where the animal could have built a prediction, right? So we had experiments I didn't go over, but we had trained a monkey to hit a little lever, and every time he hit the lever, he'd move the same way. And we did that over and over and over again. And, the neurons, always treated that as if it was a passive stimulus, right? So we thought, okay, we'll try another protocol, right? Because that seemed, very Pavlovian, very, very simple, right? Easy to make a prediction. Where we gave, some of the people probably know this experiment where we gave the monkey a steering wheel and had him steer. And the same issue, like, that experiment, we never saw any suppression. So, and you could probably come up with other variants of that, but it looks like it's consistent. I think it depends again on what the behavior is, right? And you can drive the behavior, what is the function of the pathway. So for example, if you're looking at the anterior vermis vestibular nuclei vestigial loop, that's involved in posture, right? And it makes sense for postural control. You're going to want to do that from point of, so if you came up with a state dependency within postural control, it may make sense. But if you're coming up with other types of predictions, it's not necessarily going to update for those. Does that make sense? It's not just a, it's not just a black box that's making predictions.
Sure.
It's making predictions for a particular. So we've been looking at some other perturbations we could potentially do. We actually had a whole series of experiments where we were trying to look at visual, kind of biasing things with visual input, but we never found any ability to do that in this part of the circuit.
Yeah. I'm wondering if, again, I think that that would require the execution of complex behaviors that don't rely upon an animal seated, in a monkey chair. So even in this, even in the context that you've been looking at.
Right. But we've got, we're now looking at, we have posture platforms
Sure.
and walking monkeys. So, and again, for walking, I don't necessarily predict it'll be 100% suppressed, because based on work people have done with galvanic stimulation, there are probably going to be some sort of, postural control when the monkey translates or transitions to walking. And then there seems to be some cycle dependency within swing and stance that might be revealed.
Yeah. Good. Thank you.
Yep.
We have two more questions and 5 minutes. So, Rob, you want to?
Yep.
First.
Okay, yeah, thank you, Kathy. That was a great presentation. I've been reading your papers recently, and so you explained everything much more succinctly. But so my question is,
Maybe too
you talk about what it might relate to Soroush's the suppression of the vestibular only neurons when the organism wants to do active movement, and it coincides with suppression of reflexes. So it's actual switch in the pathway. Now, you said this was very marked in one of your papers, down to like 80% of the firing rate. Yeah. And so my question is,
it's more like 70 on average.
70, okay, yeah. So it didn't appear you knew the mechanism for that.
Well, that's what we're working on, right? So we know that the vestibular nuclei gets input from anterior vermis, for example. So we've been, and that's what I was sort of went through very quickly at the end. We've been recording for the Purkinje cells where they project down to, right? So they're the logical place for that cancellation signal to come from is the cerebellum, right? And the cerebellum has been implicated in internal models for decades. So what we currently have a paper under review, where we actually do see motor inputs. We can record from Purkinje cells. We see that they have neck motor inputs, for example, right? And that they have proprioceptive inputs and they have vestibular inputs, and we can add them in a population model, simple population model, and actually produce the responses of the neurons we see either in the vestigial nucleus or in the vestibular only neurons.
Right. Well, my question is it at all possible that any of that is coming from the periphery, turning off of the irregular afferents by the efferent system, maybe?
No, that's what I was trying to make the case in the beginning of the second question of my talk, is we've looked during active movements, right? So during active versus passive movements, the afferents are they're like a MEM sensor, which is good, right? Because you don't want to, the afferents are coming in and they're targeting both PVP neurons that generate the VOR and central neurons, VO neurons that are vestibular spinal pathway. So mucking things up in the periphery has never seemed like an elegant solution to me, because you still want to drive a VOR, right? You don't want to be turning off the afferents, and it's not like all regulars go to the VOR and all irregulars go to the vestibular spinal pathways, right? It's more, it's biology, right? It's not binary. So
okay, yeah.
Right. And we did that. Like we had the monkeys generating active head movements. They're encoded exactly the same if we do replay, by moving the vestibular turntable, as the neuron doesn't signal that it's being moved one way versus the other. And we also did the walking experiments that came out in that recent Nature Communications paper. The neurons, even during walking and running, the afferents encode information as if it's passive. And
Okay.
again, it's a useful thing if you're designing a prosthetic, because if it wasn't the case, designing a prosthetic becomes much harder, right? Then you right, your mapping would have to be different if you're generating your own movement versus somebody's moving you, which would be really tricky.
Okay.
I guess.
But it does raise, yeah. So the question is, what are the efferents doing at the periphery? Because they're receiving some information.
Yeah, well, that's a big question. That's like my part-time hobby is like what I didn't talk about. And Soroush has been involved, and we've been trying to sort out for years what the efferents do. But maybe Soroush has better ideas than I do at this point.
I have some ideas. Maybe another time. Yoshiko for last question.
Thank you, Kathy, for the great talk as always. So my question is about rostral fastigial nucleus neurons activity. So at the beginning of adaptation, the head movement, I mean, the firing was too low, like head movement was too low to make the appropriate movement. But the catch-up trial, the head is too large, so the head need to be smaller. I mean, the movement need to be smaller. So I kind of expect the neurons firing differently, like make too large or make too small. So but the firing was both increased. So I'm wondering what kind of signal is this and how does that signal help the learning?
It's very simple what they're doing. So they're encoding, if there's a mismatch, they encode the head motions. That's all. So during during the catch trials, because there's a big mismatch, they just encode the movement as if it's passive. That's all. So you can think of them as they're basically shaping, they're shaped by whether or not there's a match between what the animal did and what what its expectation is based on what it did and what actual sensory feedback it gets. If there's a mismatch, it's just a kind of binary thing, though we're trying, again, as I said in the very end, understand better how that comparison, the rules of that comparison. But if there's a mismatch, it's just that it's simple. The head motion is encoded as if it's passive. So during these big catch trials, the animal generates these huge head movements and they're encoded as if they're passive. So you get these huge fire rates. Does that make sense? So it's not so it's consistent with a sensory prediction error being generated, but it's not a sensory prediction signal, right?
So it's not really error signal, like how different it was. It's
no,
more.
It just, but what it demonstrates
it
demonstrates that an error signal must be generated somewhere, right? Because somehow you need to make if you're generating a comparison, right? There's a comparison being made, right?
Okay, thank you very much.
Yep.
Thank you very much again, Kathy. Thanks everyone for staying. We went over time. Thank you so much.
Thank you for everyone. And I'm happy to follow up with any questions people have.